Hey guys, I realize you're all kind of freaking out about the presentation that you're supposed to be working on for today. Don't worry about it. It's not that big a deal. You'll be fine. I know that's kind of my mantra, but I'm almost always right too when I say that. There's going to be situations in AP or in biology in general where the artwork is what is throwing you. You need to pay attention to what the thing is doing or what it can do, and not worry about the actual way it's depicted in a drawing, because these are molecules, and an artist is going to be able to take some liberties with it because we don't really see molecules. So just do your best. Today I'll be meeting with you to answer any questions you have about any of this or anything else for that matter. So we're going to go into mutations now. So mutation is a change in DNA, and it normally with this PowerPoint, the first slide would say a mutation is a change in DNA, and then it said, every time the word mutation appears in this PowerPoint, turn to your neighbor, look them in the eye, and say a mutation is a change in DNA. Because mutation is one of those words that you need to define whenever you use it in an AP for your response. Remember, if you say a word that sounds sciencey and you don't define it, you may be missing points on a rubric. So make sure you define any sciencey word. Act like you're writing your free response to your younger sibling who does not know science very well. So if you're going to say mutation, you should always say, if nothing else, just in parentheses, a change in DNA. When we change the DNA sequence, just any random change to the DNA sequence, there are three possibilities. One, and the most common one, is that it doesn't do anything at all. Like I said, 1.5% of the genome codes for proteins. There's another big chunk of it that codes for regulatory RNAs, which is actually a bigger portion than the part that codes for genes. But the vast majority of it doesn't do anything. It's just, remnants of old genes. It's, garbage from way back when. Right? We've been passing this molecule down for, 3.5 billion years. It's got some junk in it. It's got a lot of junk in it. The second most common outcome of having something change in the DNA, having some sequence change, is that something harmful happens. It could do something harmful like cause a genetic disorder, or it could do something harmful like cause cancer. The third and by far most rare outcome of a change in DNA, of a mutation, is that it actually gives you some new trait, some new beneficial trait. So no effect, harmful, or beneficial, right? And in that order, and with big gaps in between the likelihoods. But we rely on this mutation rate that does almost nothing, and when it does do something, it's usually bad. But every once in a while, it conveys some new trait that allows you to survive and reproduce better. And that's the part that is required for evolution. And that's why you may have heard me say that, cancer and genetic disorders, these are just side effects of evolution, right? They are there because we have to have a mutation rate to maintain the diversity of our populations so that we may evolve and get new traits. All right, but anyway, let's look at a mutation and how it happens and some of the terminology that goes around it. It's pretty straightforward. So there's a variety of different types of mutations. And the simplest type is called a point mutation. That's just when you take an A and it becomes a C, or a G gets turned into a T or whatever. And this usually happens during replication. The DNA polymerase will be building, accidentally stick in the wrong nucleotide. This is a molecule that does this, thousands of these per second. So it's very, very fast. It's going to make some mistakes from time to time. There are a few possible outcomes for a point mutation. If an A gets turned into a C, it's very likely that you're going to have the wrong amino acid at that position in the final protein. And that may or may not be problematic. But it's possible that it doesn't change the amino acid at all. Remember that there were like 6 different codes that code for leucine. If you change that third nucleotide from an A to a G, in many cases, that's still going to be the same amino acid. But you could end up with the wrong amino acid. Or worst case for a point mutation is that you turn a code for an amino acid into a stop codon. And so suddenly you don't make the whole rest of the protein. So no change is a possibility. A missense mutation is when you've got the wrong amino acid. And they'll ask you this sometimes in AP. They will say, here's a series of amino acids. It's supposed to be this, this, this. And instead it's this, this, and this third one is something else. And then you would have to explain. You're not going to get away with just saying it's a missense mutation. You're going to have to explain one nucleotide got replaced with another so that we got the wrong amino acid. But that is called a missense, but never just say missense mutation. You must explain it. And a nonsense mutation, and again, you shouldn't just call it a nonsense mutation. A nonsense mutation is that situation where you turn what's supposed to be an amino acid into a stop, and it terminates the translation of the protein. Here's an example of each one. So I've got this is my sequence. Now, this is way smaller than basically every protein would be. We've only got 4 amino acids in this little protein. But so if we get a base pair substitution and it turns this C gets turned into a U, we still get glycine. We had glycine, we got glycine. And so it doesn't change anything. So that, yes, technically it's a mutation, but it's a silent one and does nothing. A missense mutation is if we change this G into an A, then suddenly we get serine instead of glycine. We've got a different amino acid. That's a missense. And then if we got a U here instead of the A here, instead of getting a lysine, we get UAG, which is a stop codon, and we stop doing translation there, and our protein is 1 amino acid long. All right, no change, missense, nonsense. Doesn't matter if you know what the name is. What matters is you understand how we got it and what it's going to cause. So when we're talking about mutations, we want to make sure we're thinking about what heritable mutations are. So take a minute, pause the video, and see if you can figure out the answer to this question. So hopefully you figured out this is going to happen in sperm, eggs, or in the zygote. Or it can happen in the cells that are going to be used to generate sperm or eggs, those precursor cells. Either way, it's got to be in the germline. It's got to be in there so that it ends up in all the cells of the offspring. That's how you're going to get a heritable mutation. And those are called germline mutations. Mutations that happen, say, in your skin or in your liver, which are common, right? Those happen all the time. Those are just called somatic. And the worst consequences of those are cancer. So germline mutations are responsible for genetic disorders or new traits. Somatic mutations, cancer or nothing really is what you get. Because it's only one cell. And so it's probably not going to have much of an effect other than if it becomes, cancerous. Now, you may remember this from freshman year, but imagine a mutation where a single nucleotide is added to the DNA. So I've got my stretch of DNA, I've got my reading frame, and I insert 1 nucleotide into there. And if I insert 1 nucleotide in there, then suddenly those 3 base pair codons that I'm reading, they got shifted. I'm going to be reading the wrong ones. So it's going to shift that reading frame. And that's called a frameshift mutation. And we can get this same type of mutation. A frameshift mutation can also be caused by a deletion, of course. But you can have insertions and deletions that don't cause a frameshift mutation. Maybe you should pause the video here and see if you can figure out what kind of insertion or deletion would not cause a frameshift mutation. So hopefully you came up with any insertion or deletion that it comes in a multiple of 3 nucleotides, right? Because if there's 3 nucleotides, then your reading frame is still the same. It's just, if it's an insertion of 3, now you've got 1 more amino acid than what you should have had, but the rest of the amino acids are all the right ones. If I had just an insertion of 1, then every amino acid is wrong after that. If I had an insertion of 2, every amino acid is wrong after that. But if I have an insertion of 3, then everyone's still going to be right. There's just going to be 1 additional amino acid. And the same thing's true with deletions. If I have a deletion of 1, everything's wrong after that. A deletion of 2, everything's wrong after that. But if I have a deletion of 3, everything's right. I'm just missing 1 amino acid because every 3, right, codes for a single amino acid. So here we are with our little bitty protein again. If we are missing this 3rd U right here, so that 3rd uracil is missing, and so we end up with the wrong amino acid. Instead of phenylalanine, we got leucine here, and we didn't get glycine, we got alanine, and we didn't stop. We keep on going. So we're going to get a protein that's not even the appropriate size, because who knows where we're going to stop. It is possible also by inserting, we could cause a nonsense mutation with an insertion or a deletion if we make a stop codon early. And then this last one just shows that if we insert or delete 3 nucleotides, right? This is a deletion of 3 nucleotides. We're missing this AAG, and we're missing lysine. So our protein is only 3 amino acids. Now, it's maybe more likely that the deletion would be, something overlapping a couple of different ones, but you would still only be missing 1 amino acid. And this one would be wrong, but then the rest of them would be fine after that. With a frameshift mutation, if that happens early in the protein, it's much more devastating than if it happens at the end. In fact, it may be okay if it's right near the stop codon. If it's near the very end, we may be okay because we may be making a part of the protein that is not part of the active site, and the protein may work fine. So when we look at sickle cell anemia, the wild type or normal version of hemoglobin, the most common variant, has this one sequence that says CTT, and it codes for a glutamic acid. In the mutant version, you have CAT, which codes for valine, a different amino acid. So that is, of course, a missense mutation. And of course, it's a point mutation. Thank you, penguin. Now, there are a variety of mutations that cause cystic fibrosis, but the most common one, and I think I mentioned this in class, the most common one is a 3-nucleotide deletion, which is really interesting because it's a rare mutation. It's a very specific mutation, but it's a deletion, but it does not cause a frame shift because it's a 3-nucleotide deletion. Another source of mutations is caused by transposons. Hey, Jen, who's the transposon lady? Jumping genes lady? Barbara McClintock. Transposons were discovered using corn by Barbara McClintock here. And they're really interesting, and they are everywhere in everybody's genome. And they're little pieces of DNA that make copies of themselves and then insert those copies elsewhere in the DNA. So they act like a little virus, but they never leave the cell. They just do it within your own set of DNA. It's a strange little gene. But these things are everywhere in our genome. In fact, this is a significant portion of our genome are just copies of these little jumping genes. Now, the problems that happen is if this jumping gene jumps into the middle of one of our normal genes, then we're going to disrupt the function. Now, that's only going to be really a problem if that happens in, a germline cell, in a cell that's going to make a whole organism, because now you've lost the function of that gene. But this is a common mutation. So if you look at this transposable element, so jumping genes, transposons, transposable elements, all the same thing. So I look at this jumping gene, and let's say it gets copied and inserted in different places. Let's look at what the effect is. So if it jumps right into the middle of an exon and a place that's going to be coded for a protein, that's probably going to destroy the function of that protein because this sequence is supposed to be here. It may cause a frame shift at this point, but it's certainly going to make a big chunk of protein that is not supposed to be there and probably disrupt its function. You could also screw up the promoter sequence. And if that's the case, then you may end up making it so the gene is not expressed, making it so the gene is expressed in the wrong places. All kinds of problems can come from that. If it jumps elsewhere and it's not in a gene, it's probably not going to cause any problems at all. It's not disrupting anything that we use, so no problems there. So our genome is about 44% transposons. That's a big chunk of our genome that is just this copies. But think about how these things make this themselves, and it makes a lot of sense that these are going to be, that they're common. Oh, and over 10% of our genome is actually the same insertion repeated here and there all throughout our genome. Because this little guy gets busy and starts jumping and putting copies of itself elsewhere in our genome. 10% of our genome, a single rogue gene. We also need to talk about SNPs. If you do like 23andMe or any of these genetic tests that you can mail in, what they're going to look at is your SNPs, because SNPs are associated with specific phenotypes. They're associated with specific historical populations. And so we can use these for various types of identification. Now, most of the stuff between you and the person next to you, well, the person next to you is probably a family member. The person who used to be next to you when we sat in the classroom, most of those differences are not in the genes. They're in how you express the genes. A few are. There are variations within genes, but for the most part, you're pretty similar. But there are specific locations in the genome that vary more often than others. So a SNP is a single nucleotide polymorphism. It's a single nucleotide location that varies in at least 1% of the human population. And so if, in a lot of locations, everybody's got a T in this spot, right? Everybody's got an A in this spot. But if more than 1% of the population has a difference at a particular location, then that particular location is called a SNP. And a haplotype is a collection of SNPs. So in an actual physical region of the chromosome, if you have a certain set of SNPs, those can be associated with genetic disorders or with certain historical populations. So you can say, oh yeah, this person's Italian because this haplotype occurs in almost all Italians. The problem with that, of course, is that humans move, and humans have always moved. And so there's a lot of mixing, and there always has been. And there's a lot of mutation, and there always has been. So SNPs and haplotypes have been associated with specific conditions. So what we do is we look at all the SNPs, we look at all these haplotypes, these combinations of SNPs, and we look for people with, say, type 2 diabetes. We look at that, and we see, oh, well, these people that have type 2 diabetes often have this haplotype on this chromosome, this haplotype on this chromosome, this haplotype on this chromosome. And that combination of SNPs indicates an increased likelihood for having type 2 diabetes. The same is true with heart conditions, cancers of various kinds, and all kinds of other conditions. Most of the things that kill us are not a single gene mutation, right? They're much more complicated than that. But we can find these correlations. So when they look at your genome, they can see, oh, you have these haplotypes that are associated with specific conditions. Of course, the value of mutations, like I said at the top of this, is diversity, right? That is the original source of all variation. Yes, they do. Okay, legacy. And my respectful response. So, mutations, changes in DNA, don't forget to say it. Make sure you understand the way mutations can happen. Make sure you understand how they're going to affect the protein. A lot of times they give you a sequence of amino acids and then they ask you, what caused this problem? And again, you can't just name it, you have to describe it. If it's a frame shift caused by an insertion or deletion, you would say an insertion or deletion of a nucleotide or two would cause all of these amino acids to change after this particular point. Something like that. Talk to you guys soon. Hang in there. You're doing great.